This is definitely not my favorite category. So we have to understand a little bit about electricity, but in the body, we don't use the same words that they would use in a physics class. So the first thing to understand is that we're talking about electricity, and one of the ways we can measure electricity is voltage. But in the body, we don't call it voltage. We don't say that like you're electrified and you have 120 volts. What we say is that you have potential. And the reason we use that word is because voltage has the potential to move ions from one place to another. So it's kind of like a force that we can use to move ions from one place to another if needed. So potential means voltage, and we're going to start just using potential. And when there is potential or voltage, there's a difference in the distribution of ions. And you're thinking to yourself, that's a concentration gradient. It is, but this is a concentration gradient of positives and negatives. So take a look at this cell over here. Like you can draw something like this cell if you wanted to, right? You can do a circle and you can say that there's lots of positives on the outside of the cell because that's true. There's lots of sodium on the outside of the cell. And I know you're going to say, but Amy, there's lots of potassium on the inside of the cell. Yeah, but there's also these really big proteins. And those are very negative. And they really make the inside of the cell negative. So getting back to our definition up top, there's a difference in ion distribution. And there's the ability to move ions. This is the same thing like two terminals on a 9-volt battery. They have a difference in ion distribution. There is the ability to move ions. Okay, so we're understanding that cells are charged, kind of like a 9-volt battery, but we don't say you have 9 volts. We say that your cells have a resting potential. I want you to think about that statement again. A battery is measured in voltage, and you are measured in resting potential. What the heck is going on in this diagram, right? Well, okay, so the idea here is that chemical gradients exist, right? And you already know that. You know that there's lots of potassium on the inside of the cell, which is orange in your book. And you also know that there's lots of sodium on the outside of the cell, which is usually like a steely blue in your book. So chemical gradients exist. And we already know that. The second thing is that electrical gradients exist. And we just established that. We said that there's lots of positives on the outside of the cell. You can see them here. And we said that there's lots of negatives on the inside of the cell because all of these big proteins that are really negative. Okay, so the other thing, there's another force that's actually determining your voltage or your resting potential. And that force is the sodium-potassium pump. And we know that that pump uses ATP to move ions across the cell membrane. Okay, so all of these things contribute to what is called your resting potential. So a cell has voltage. This is why. Here's 3 reasons. And we measure that voltage as a resting potential. So when the cell is just kind of hanging out, all of these forces are active, but they balance out. And your cells, if they were a little tiny battery, they would have a voltage of negative 70 millivolts, usually, around. Okay, now that we understand that cells have voltage and that that's called a potential, we're going to start calling those membrane potentials because it really is about moving ions across the membrane. There are 3 types of potentials. There's the resting potential, which we just established. We gave 3 reasons for why it exists: chemical gradients, electrical gradients, and the NAK pump. And we know that it's usually, we have a number for it, it's usually around negative 70. We need to understand that there are 2 other types of potentials. Graded potentials, which is a change in voltage or potential that dies out. It's like a match. It lights but it dies out. An action potential is kind of like a fuse to dynamite. You light it and it goes all the way to the end. What does this mean for a neuron? Well, remember our neuron structure here. And we defined this area here as the axon hillock, right? And so that's the axon hillock. And then this big long cable here is the axon. And actually, the axon hillock acts as like a resistance to an action potential. So over here in the cell, we get an action potential and it goes, but it can't go any further. And that's a graded potential. Over here, we get an action potential in the cell and it gets to here and it goes, yeah, and it goes all the way down the axon and lights another one in the other cell. And that's an action potential. So don't be confused. There are different types of potentials. Be aware of something. It's knowledge that we've picked up along the way. It just might not be something that you've consolidated here. Be aware of something that in a cell, it has channels in the membrane that are always open. So it's like cells are leaky, and they always are. And so sodium constantly leaks into the cell and potassium constantly leaks out. So that's one type of channel where there's free movement across and back. There are also chemically gated channels, and this is like crossing a bridge where you have to put a token in, or entering a hotel room where you have to use a key. In the past, these have been called ligand-gated channels. We talked about one of these when we talked about acetylcholine opening up channels on a muscle fiber at the neuromuscular junction. That's like using a key, a physical molecule, to open up the channel. All right, so we have channels that are leaky, channels that open up with a molecule, and then we have channels that open up with a change in voltage. This is much like the key cards that are used in hotels these days. There's basically a line of voltage, and you break it by inserting your key card, and that's what opens the door. This is very similar to a voltage-gated channel. So just be aware that there are different ways that you can move through a cell's membrane, and these are some of the different ways. You have free movement, you need a key, you need some voltage. I don't like the pictures in your book at all. They make like no sense to me. So let's just understand what a graded potential is. So it's a local change. That means that it's restricted to a very small part of the membrane in the neuron. And you can see that in these pictures over on the left. They're showing you really, really small parts in the cell body of the neuron. So what happens is that gated channels, like the sodium channels and potassium channels, they open up from a stimulus. So this neuron receives a stimulus from something in the environment, and some of the channels open up, but not enough of them opens up for the potential to change all throughout the membrane, right? Remember that axon hillock prevents them from going any further. So they get a change in voltage or potential, but they just can't get any further than right there. Oh, I said that. Okay, what is a postsynaptic potential? Think of yourself as a neuron, and you can be excited in different ways, and you are irritated and excited in different ways all the time. So you're at your resting potential, but sometimes you receive a stimulus and you go up from that, or sometimes you go down from that. And these are called postsynaptic potentials. They're graded potentials in the postsynaptic neuron. So let's draw this. Here's a presynaptic neuron. Here's our synapse, the space between the two, right? So this is the presynaptic neuron. This is the postsynaptic neuron. And what we're saying is that this neuron here, the postsynaptic, can have graded potentials to make it either more willing to be excited by the presynaptic neuron or less willing to be excited by the presynaptic neuron. So these would increase the ability to have an action potential or decrease. Think about that for a minute. That's anesthesia, decreasing the likelihood of an action potential. So we have two kinds of postsynaptic potentials. And the first type is an excitatory postsynaptic potential. That's what EPSP is. This means that the postsynaptic cell really, really wants an action potential. Can't generate one itself, but it's like, yes, 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 please. And this means that it's moving more toward threshold, but it can't get there on its own. But it's really excited, and it wants an action potential. And if even a little bit of potential change comes its way, it will have an action potential. The postsynaptic cell can also be inhibited by graded potentials. This is like anesthesia that makes muscle contractions and thoughts less likely to happen. This moves away from threshold, right? This makes the action potential less likely. This is the postsynaptic cell saying, no, 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 I'm not having action potential. And in fact, I'm so adverse to it, I'm going to reduce my voltage. There are many ways to get one graded potential to become an action potential. Many, many ways for that to happen. And the reality is that one neuron does not usually connect to one neuron. One neuron is usually connected to many. One cell body, like the ones that are pictured here, can have many attachments to it from presynaptic cells. So let's just consider one way that you can change a graded potential to an action potential. Let's say that one graded potential is not enough. It would take two. We have to have two graded potentials, and that will help the cell reach threshold. There's two ways that we can do that. We can have two graded potentials arrive at the same time, and that's what you see pictured here. This neuron is being stimulated by two other neurons at this site, and that's enough to get us over the axon hillock and propagate an action potential. So that's one way, and that makes sense, right? The other way is called temporal, and this means two action potentials in succession of each other very, very quickly. So this picture here is kind of, I mean, it's not bad, but so what we're saying is that there's one arriving stimulus and then a second one really quickly, right? So the two of them arriving very quickly on top of each other, those create an action potential and propagate the action potential down the axon hillock. So when you think about it, there's two ways, right? Two at the same time, or two very, very quickly in succession. Okay, so we've defined graded potentials, right? And graded potentials, they irritate a membrane, changing its voltage, but the voltage dies out. But what does change a graded potential to an action potential? There is a significant point at which you change a graded potential to an action potential. You have to generate enough voltage or change in voltage to do this. If you reach this point, which is called threshold, then all of the steps of an action potential will proceed. There's no like one step and then, oh, sorry, and then you stop. It's you reach this threshold and a graded potential becomes an action potential. All right, I understand the concept of a threshold, but what is it? It's a voltage. Your cell is hanging out at -70. That's its resting potential. Something irritates it, and that voltage rises, right? That potential rises. That's what's going on here. Cell is hanging out, something irritates it, and it's like, eh. And a graded potential will just go, and go back to the resting potential and be done with it. But an action potential reaches this voltage and proceeds with all of its steps. So I've drawn an action potential here in blue, and I've drawn a graded potential here in yellow. These graded potentials are usually confused with the resting potential, which is one, remember that's one value on this y-axis here, right? So the resting potential is where the cell is chilling out at. So just be aware. You've established some terms throughout this mini lecture. I don't know if you realize that, but these are terms that we've defined before. We've used them. We've used this term depolarization to become more positive, right? So a cell is hanging out at its resting potential, which is noted down here at -70. And the cell is more likely to have an action potential. That's depolarization. It's moving more toward the threshold, which is probably somewhere up here. Depolarization means that the cell is becoming more positive. And this means that sodium is rushing into the cell and sodium channels have been opened. Repolarization means that the cell becomes more negative. And this is when potassium channels have opened and potassium is leaving the cell. And you might say, wait, but that would make it more positive. No. Potassiums are positive. If they leave, the cell will become more negative. There is another word that we apply, and it's called hyperpolarization. And this is to become more negative, below the resting potential. Look at how repolarization is to become more negative above the resting potential. Hyperpolarization is below the resting potential. So many students will start to understand action potentials and they'll be like, wait a minute. When do we use these different terms? They seem to be the same thing. Yes, but we're talking about them in relationship to the resting potential.